seminar. And, you know, we're going to keep, like I was just saying, we're going to keep doing this for quite a while. So, you know, stay tuned every Thursday. But this Thursday, I'm happy to introduce Hannah Yevick, who's local. She's over at MIT, luckily for us. And she has like a very broad background in physics, you know, starting with her bachelor's at Penn and then moving on to Columbia and then doing her PhD at the Curie Institute in Paris. And she's like published on things from graphene to cell mechanics, right? So, but today she's going to talk about sort of that sort of latest thing about how cells interact in embryos and how those mechanical interactions like lead to sort of robust structures that you can generate. So with that, the other side of this is that, you know, everyone's muted when you enter, right? So if you have a question, you can do a couple things, right? You can send a chat and I can read that for you, or you can raise your hand and I can call on you. Or if you are really excited, you can just unmute yourself and ask a question. Hopefully I'm like that. And we really, that's important for you to kind of ask questions because Hannah's going to be talking to her computer screen the whole time. And it's going to be really awkward if you don't ask questions. So like, just, you know, say hello. All right. So, you know, I'm going to mute myself now, but Hannah, take it away.
Okay. Well, thanks so much for the introduction and for the invitation to speak about what I've been doing, work I've been doing on the mechanics of robust tissue folding. So what I'm interested in is the physics of how complex 3D shape is established during development. And namely, how do you go from a single cell or sphere into a complex multicellular organism, which is comprised of many different intricate structures. And so this process, called morphogenesis, requires the coordinated movement and deformation of huge populations of cells. And so my work addresses the physics of how simple interactions between cells in a tissue can drive the necessary shape changes robustly needed for tissue development. So this question of morphogenesis has been the fascination of biologists for a long time, and huge advances have been made on our understanding of genetic and molecular players involved in development. So for example, which genes are needed to be turned on to control specific phenotypes or observable traits of an organism, or how cells at specific positions in an embryo know which fate to go on and become. So what I'm showing you here, I think, is a really beautiful example of this. We're seeing a fruit fly embryo, which is a system I'll be using later in my talk. Cell nuclei are in green, and two genes patterns which are being expressed are shown here in blue and red. And so it's not really important what these genes are doing here, but I want to show is that there's these precise patterns of gene expression in an embryo that can relay positional information to specific cells to tell them what structures to go on and form. So while we have sort of these gene descriptions, ultimately these are sort of static pictures of development that have huge limitations because if we look at a developing embryo, development isn't just a biochemical process, but it's also a dynamic mechanical one. So cells, as you can see here, they must like move, deform, and exert forces on their neighbors to build the global shapes that are needed inside an organism. So a big gap in our understanding is how do you go from these gene patterns to the collective movements in target structures. And so this is where physics can come in to provide really a whole different set of perspectives and techniques to understand this important biological question. So one natural framework to describe these emergent, these emergent population level patterns is active matter. And so active matter, as I'm sure you're familiar with, systems are composed of a large number of self-propelled units, each which consumes energy to exert force and move. And so by introducing simple interaction rules between units in your system, you can explain the similar physics can explain how sheep flock or schools of fish swim together, or even at completely different length scales, how sort of collective patterns are formed inside tissues or groups of cells. And an example of this is seen here on the left with cells, kidney cells, which are seated on a flat dish under a microscope. And so for the interest of this talk, what I want to highlight is that in all of these systems, you don't actually need a specific leader to tell the others what to do and what pattern to go on and form and how to do it. But rather what we have here is that simply through the interactions of individuals, the entire system can establish these highly robust global patterns. So what I'm interested in is can local interaction rules between cells in a tissue explain some of the robust movements that we see during morphogenesis as tissues build their shape. So I'd like to argue that this question is an exciting one for those of you here interested in bio-inspired materials, because tissues can ultimately just be thought of as a soft material. And so what I want to understand is how does this material self-organize and robustly grow into a final target structure almost every time, while still maintaining the ability to do really fascinating things like adapt and reconfigure to different environments, grow, self-heal, or move. And clearly from a materials engineering standpoint, this would be a really highly enviable and enticing system to know how to program and build. And so learning rules of how tissues control their assembly can ultimately provide us with a framework to think about designing self-organizing materials outside of living organisms. So when thinking about a tissue as a soft material, diverse models and concepts from soft condensed matter physics have been successful in describing material properties of tissues in different contexts, where in some situations, tissues are better described as liquid crystals, in other cases, foams, or even as granular material. But what's exciting is that tissues themselves are active, and so they're inherently out of equilibrium. And so there's been they really present a playground for thinking about new and interesting out of equilibrium physics. And so, for example, in the case where cells organize, or cells and tissues organize like liquid crystals with long range order, it's been seen that you can have the creation of orientational defects inside your system over time, which is counter to what would be expected in a passive liquid crystal, which would relax towards an equilibrium state. In cases where cells organize more like foams, it's been observed, there's been observed interesting packing of cells that you wouldn't expect in an equilibrium configuration or novel neighbor exchanges. And finally, in cases where cells organize more like a granular material, they can undergo jamming and unjamming depending on throughout the course of development.
Can I ask a quick question?
Sure.
Is there anything general about when a tissue is more modeled by one of these active liquid crystal models or foams or this jamming thing?
Well, I don't know if there's like, is there a unified description of like when falls in? I think,
or just like a few examples. I don't know.
I think it depends on the context. I mean, to be honest, some tissues that people have described as more jamming, unjamming transitions can also be thought of in, as foams as well. So they're, you know, it's not specifically granular material. In terms of when cells organize more as liquid crystals, it's usually because cells themselves exhibit a highly elongated body, because that's sort of just the cell type that it is. But they're a little bit interchangeable depending on the phenomenon you're trying to describe. I would say that. I don't know if that helps.
No, it does. Thank you.
So yeah, but so while these descriptions capture some elements of how cells interact in a tissue, the challenge is to go from these somewhat simplified 2D descriptions to understanding 3D shape, which, as you can imagine, introduces new degrees of freedom and complexity in your model. And so, as you can imagine, why this is important is because tissues are organized into highly curved 3D structures with diverse shape in vivo. And so here are just some striking examples, as the case in the salivary gland, the lung, the kidney, or the breast, where you can see completely different structures that form. So one way to achieve 3D structure is through the folding of a 2D sheet. So this is illustrated here, where you can take a simple dollar bill, and by applying creases over the course of time, you can build this final 3D structure. But as you can imagine, by varying the position and timing of these folds, we can end up with completely different final outcomes. And so this strategy of folding to formed 3D shape is used, in fact, during development, where in tissue folding builds completely different structures during development. So for example, the optic cup, which would go on to form the eye, folding of the neural tube, which forms the central nervous system, and the folding of the ventral furrow in the early embryo, which goes on to define cells that will become muscle. And then I just illustrated here, this occurs also across different organisms, as I'm illustrating in the zebrafish, the mouse, or the fruit fly. And later in my talk, I'll be describing a bit more this fruit fly ventral furrow tissue fold. So how do cells interact to sculpt curved tissues? And as you can imagine, to decipher the patterns of cell interactions in vivo and in vivo tissues is very complex, as there are usually many simultaneous variables. So for example, cells in a tissue are dividing, shape is evolving, as well as complicating external factors such as movement of neighboring tissues and so on. So to really try and parse out sort of how simple rule of how cells are interacting, you need to define ways to deconstruct this complexity. And so there's two different strategies that you can imagine. One would be a bottom-up approach, where you can isolate individual variables in a system, for example, using engineered microenvironments. So for example, if you want to understand how population size or the number of cells in your tissue impacts the way they coordinate, you can put a certain number of cells inside a dish and then scale up and see how that one variable impacts collective or emergent properties. An alternative approach would be top-down, where you can try and find the most simple developing tissue or a simple context of a developing tissue, and then use quantitative analysis to try and parse out some of these rules of how cells are coordinating. And so today I'll tell you about two different projects where I used exactly these techniques. So first, using a bottom-up approach to address how out-of-plane curvature guides cell interactions. And then second, using a top-down approach where I looked at a simple folding tissue in the fruit fly embryo to identify how cell interactions robustly establish tissue curvature. So this first part of my talk was done at the Curie Institute in Paris in the lab of Pascal Silleberzen. And so this was motivated by the fact that I described before, that physical models of collective behaviors in tissue were largely limited to 2 dimensions, but we never actually have these flat ideal conditions in vivo. And so while I showed before that cells organize into highly curved structures, they also are constantly confronted with highly curved environments when they move in vivo. So just an example is shown here during cancer invasion, where cells are escaping a primary tumor. So what we're seeing is cell strands moving collectively, where cell nuclei are yellow and the cytoplasm are red. And we're looking at 3 different Z heights of muscle fibers shown in gray. And so these muscle fibers are forming highly curved tracks that the cells are forced to move along as they metastasize and try and find a second location in the body to form a secondary tumor. So the first thing I was interested in is, how does curvature influence a cell's ability, or a population of cells' ability, to move together? So, my goal was to systematically introduce curvature into a flat system to isolate the impact of curvature on a tissue. So to do so, I used a glass wire assay by seeding cells on the outside of glass wires of decreasing radius. So as you can imagine, on large enough glass wires, cells that are grown on the outside of large enough glass wires are in a nearly flat condition, and then you can tune in out of plane curvature into your tissue. But as you can see, when cells are grown on the outside of a cylinder, they don't just experience out of plane curvature, but ultimately they also experience lateral confinement around the circumference. So to decouple the effects of confinement from curvature, I systematically compared my results to cells confined into micropatterned tracks on a glass slide of decreasing width. Okay, so what does this look like? As you can see here, cells are seated on a support, and then when the tissue is confluent enough or dense enough, they begin to move down. This is a glass wire in this migrating front. And the cells, as you can see here, migrate collectively down the wire, allowing us to ask the question of how migration speed is impacted by wire radius. So I've plotted this here, where I'm plotting this collective migration speed, or of the front, with respect to the lateral confinement for both cell tissues confined into wires or tracks. And so if we first focus on the black points, or the wires, you can see that below a lateral confinement of around 200 microns, which is just around a radius of 40 microns, the migrating front starts to speed up and goes faster. But ultimately, velocity, as you can see now looking at the red points, is dictated by confinement and not curvature. So how do we understand this increase in speed of the migrating front below around 200 microns? This can actually be understood by going back and looking at these collective swirling patterns that I had showed you before on a previous slide. And so what happens is that if we look at the velocity correlation length of cells in this tissue, it's around 200 microns, meaning that these sort of collective swirls form at around that, in patches of around that size. And so if we confine our tissue basically below that width of 200 microns, we're basically suppressing these swirls and causing the overall tissue to stream down the track or the wire in a more directed fashion. So if the speed of the migrating front, or the collective migrating front, is not controlled by curvature, but rather by lateral confinement, what we identified is that there's actually a transition from collective to single cell migration, which is in fact driven by curvature. So this is an example here on a wire of 5 microns, where cells break away from this collectively migrating front and move up and down the wire at very high velocities. And this is just quantified for you here, where the frequency of detachment, or these sort of breakage events, becomes increasingly frequent at smaller and smaller radius wires. And so this is surprising because the cell type that I'm using, which MDCK cells, which are an epithelial cell type, are known to be highly adhesive with each other. And so you would expect them to always move together in a collective fashion. And these types of breakage events are what occurs during cancer, where cells escape from a adhesive primary tumor to metastasize. And people have been able to identify biochemical factors that trigger these breakage events. But interestingly, what we're seeing here is that a similar, at least the way it looks, I mean, type of breakage event can be induced purely by the mechanical environment of your tissue. So this sort of surprising transition from collective to single cell migration induced by curvature got us to ask the question, how does curvature impact the internal cell structure? And so to do so, we looked at the actin cytoskeleton across the tissue, which is a mechanosensitive polymer network that helps cells maintain their shape and support the central functions, for example, cell migration or movement. And so I'm showing you here the actin cytoskeleton across multiple cells in the tissue on wires of different radius, so 85, 20, and 5. If we first look at the large enough wire, the actin cytoskeleton seems to be somewhat disorganized. But here at the smaller and smaller wires, so 20 and 5 microns, we started to see the actin cytoskeleton organizing into these strikingly aligned fibers, which then go on to connect through cell-cell junctions with aligned actin fibers in neighboring cells. And so this actin, I'll be calling it as supracellular, meaning that it's moves through cell-cell boundaries, as you see here with these white arrows, which sort of point out the cell-cell boundary. And so we wanted to quantify this. We could take the distribution of angles of these actin fibers across the whole tissue and define an order parameter of the average of cosine 2 theta, where a 0 would mean there's a random orientation of this actin cytoskeleton, -1 would be purely rings, and +1 would be the actin organizing along the long axis of the wire. And what we saw is this sharp transition between a disordered state and one where the actin cytoskeleton organizes into these striking ring patterns at around a radius of 40 microns. Comparing these results to cell tissues inside 2D tracks, we see that indeed these circumferential rings are triggered by curvature, where basically there's an alignment along the track which was induced by a boundary effect with the edge of the micropattern. So in small enough tracks, you had only one cell and that aligned with the boundary. And as your width of the track got wider, you're sort of losing that boundary effect as you average out across your tissue. So to conclude this first part, what I've shown you is that I developed a way to differentiate between confinement and curvature and identify that out of plane curvature in and of itself can cause the transition from collective to single cell migration and also induce the formation of supracellular cytoskeletal rings across your tissue. So this project, and specifically these actin rings, these beautiful supracellular actin rings, got me excited about supracellular linkages in general, and how these types of connections between cells might coordinate how they interact and therefore maybe direct global movements in the tissue. And so this motivated me to explore the next project, which I'll just tell you about, which is again about supracellular structures in a tissue, but in a completely different system, namely the developing fruit fly embryo. And what I identified is that a supracellular cytoskeletal network there also depends on global tissue properties, and that the structure in and of itself can help encode the tissue's ability to change shape in a robust manner. So the second part here is this top-down approach that I'll describe, where I identified how cell interactions robustly establish tissue curvature. And this work was done more recently in the lab of Adam Martin at MIT. So to understand how cells interact to generate tissue curvature in folded tissue, we selected the fruit fly as a model system. So the early fruit fly embryo is really an ideal model of robust tissue folding because it's driven purely by cell shape change and the resulting tissue-wide forces. So you can really isolate how this sort of pure folding event occurs without other complicating factors, like simultaneous movement of neighboring tissues or cell divisions and so on. And so what occurs is the early fruit fly embryo consists of a single epithelial sheet surrounding a central yolk, as you can kind of see in these cross sections here. And these cells are polarized, which means that they have an apical side facing outwards and a basal side facing towards the central yolk here. And about 3 hours after fertilization, you get a group of around 1,000 cells on one side of the embryo undergo apical constriction, which means that the cell apex or exterior face decreases in size, causing the cell configuration to go from sort of cylindrical to wedge-like. And then at the tissue of the scale, this triggers the tissue level bending and then overall internalization of these cells. And so what we know is that this tissue folding event is driven by the motor protein myosin, which when it's in its active conformation, it organizes into these mini filaments where motor heads are pointing outward. And these motor heads walk along anti-parallel F-actin fibers, creating a pulling force. So myosin organizes into the apex or outer surface of these cells. Whoops, okay. Myosin accumulates in the apical surface of these cells. And then as you can see here, what happens is it basically pulls on this actin meshwork, reducing that top surface and creating the wedge overall conformation of the cell. So while initially myosin sort of accumulates in somewhat of a node-like formation in the center of each cell apex, leading up to when the tissue begins to fold, it actually begins to form more of a fibrous-like structure, linking or forming a filamentous structure across cell apex, and then linking across cell-cell boundaries, as you can see here in magenta, with myosin structure in neighboring cells. And so this forms mechanical connections that link across cell-cell junctions across the entire tissue. So these supracellular myosin connections. So what does this look like at the tissue scale? So here we're looking at a folding embryo, looking down on these ventral cells that are accumulating myosin on their outside surface, where myosin is green and cell membranes in magenta. And as you can see here, the myosin forms a striking supracellular network of connections between cells, between this huge group of constricting cells, which forms sort of just prior to the tissue folding and then as it's internalizing.
Ask a quick question.
Yeah.
So how do actin or myosin go through the cell walls?
Yeah, so it's basically links through an adherence junction with an actin and myosin in the neighboring cell. The actin meshwork is more uniform in these cells, and so it's really myosin that kind of relays the force and structure, and it links and forms sort of like a mechanical link through the junction with myosin in the neighboring cell. So it's not actually, myosin can't pass into the neighboring cell. It's similar to what was, in the same idea with the previous project I described with those actin rings. The actin is still sort of sequestered in each cell separately, but it sort of forms just direct linkages through junctions.
Okay, thanks.
Yeah. Okay. So we have the supracellular network, but the challenge has really been trying to. The function of this network had really been poorly quantified and understood because you're dealing with a highly noisy image when you're thinking about imaging myosin, which is sort of a subcellular localization at the tissue scale. And so what I did is, in order to quantify myosin in these sort of highly noisy images, I adapted an algorithm that was formed to trace highly noisy filamentous structure and astronomical data to basically trace the highly noisy filamentous structure of this myosin signal across the entire tissue. And so what this does is it does a topological smoothing on your image, where basically you can then transform your myosin image into a network description, where nodes in your network correspond to peaks in signal, so sort of topological peaks, and then edges in your network correspond to the topological ridges, or sort of the mountain range peaks that connect between those two local maxima. So this, I'm just showing you in the inset here what this looks like, whereas you can see here, sort of these bright points in red correspond into these nodes in magenta, and these are your myosin nodes, and then you have the edges connecting them. So what we can now do is then map out the entire myosin network of connections across tissue and over developmental time. So the first thing we did when we could map out these connections was to ask how they were oriented, to see if there were some sort of global organization like was observed in the WIRE project I described before. So what we're seeing here is just one example of this myosin network at exactly right before at what I'll be defining as t = 0, where it's just as the tissue starts to fold, where each edge in our network is color-coded by its orientation with respect to the AP axis, or the long axis of your embryo. And just to remind you, the fold occurs here along, sort of parallel to the long axis. And what we see is that there was a bias in the orientation of edges in the network, where more edges were oriented along the AP axis compared to the dorsal ventral, or DV axis. So then we wanted to understand if, similar to wires, could global tissue geometry impact the local organization of edges in your network. So to do that, we changed the tissue geometry. So to remind you, in the wild type, we have a small rectangular patch of cells that accumulate myosin and form in a network. And we used a mutant, which basically expanded the domain of cells that express myosin and start connecting and forming this network. What we saw is that by changing tissue geometry, that we lost this bias and this increased frequency of anterior-posterior edges in our network. So the orientation and bias is lost in this mutant. And if we look at what this does to the actual tissue folding, it was striking, where normally the tissue should fold along the long axis. And what we see here is by disrupting the orientation of edges in our network, we get the tissue to fold incorrectly along the perpendicular direction. So what we're seeing here is that network properties are important for robust tissue folding. So we wanted to understand, though, more deeply, how are robust mechanics ensured during morphogenesis? So morphogenesis must be able to overcome many different types of perturbations for to be robust. So as you can imagine, a developing tissue is confronted with mechanical perturbations, genetic mutations, or other types of stochastic events on its path towards its correct tissue shape. And so, while a lot of work has been done on understanding how robustness is achieved at the genetic or protein level, it's really still poorly understood how mechanically tissues are able to respond to perturbation. So for example, loss of cells, change in cell number, localized damage, or varying protein levels, and adapt the pattern of force in the remaining cells to achieve the correct final tissue shape. And so when I was thinking about how this myosin network, or the network in the tissue, might potentially coordinate tissue scale properties and maybe the robustness of tissue folding, it got me thinking more generally about networks and network architecture, and how the way a network is organized can impact the overall robustness of a system and its resilience to damage. So here we're just seeing 3 examples of subway networks. And what's sort of intuitive is that redundancy in your network system is important to ensure its resilience under damage. So you can imagine you want to ensure that there are a lot of different backup paths in your system where if some nodes or subway stations, for example, get damaged, the rest of the system can still continue to function. So I got interested in thinking about, is this the strategy that the embryo uses and potentially is present in this supracellular network? So one way to explore or to measure redundancy in a network is through this measure called Betweenness centrality. So what Betweenness centrality does is it calculated for each node in a network, and it's the number of times a node, so for example, node B, is used on the shortest path between pairs of any 2 other nodes in your system. So for example, B is on the shortest path between A and C, A and D, and so on, and then normalized by the total number of paths. So what it basically tells you is a node with high betweenness centrality is important connecting a lot of other parts of your system together. So we can just, for example, look at this here. What I'm showing you is the node in each of these subway networks that has the highest betweenness centrality. And so as you can see, for example, in Boston, it's 0.19 for Park Street. And what that basically means is that Park Street is very essential for the whole system, and that if there's a disabled train at Park Street, basically everyone suffers. And so, you know, betterly engineered systems, so for example, in New York or Paris, maintain a high degree of redundancy, and therefore the nodes retain a relatively low betweenness centrality. So what does this look like for this supracellular network in our system? So here I'm just showing you the betweenness centrality calculated for each node in your network at one time point, which is just, again, t equals 0, just when the fold begins. And what we see is that MINDI redundant paths exist across your network. And the overall betweenness centrality in our system is relatively low. So if we look at how this evolves over time, so we look at the average betweenness centrality of all nodes in your network, and so these colors are for different embryos, and look at how that changes leading up to tissue folding, we see that the average betweenness centrality decreases, or the overall redundancy in your system increases prior to tissue folding. Then we wanted to ask, well, is there anything important about this level of redundancy in your system to actually trigger the folding event? So we tested this in 2 different ways. First, we used a genetic perturbation to basically degrade the amount of myosin in your system with varying amounts. And so we could get phenotypes where we degraded all the way down to 35% of the network's original size, so where there's very little myosin left in the tissue to connect. And what we saw is in all these different levels of myosin, we were still able to, the tissue was still able to form some minimal network, and the tissue still went on to fold and develop correctly. Another way to disrupt the level of redundancy in your system was through mechanical perturbation, where we basically damaged patches of cells in the tissue prior to which basically made those cells unable to accumulate myosin and connect into the network. And even up to ablations of 32 cells, the remaining cells were able to connect across the tissue, and the tissue continued to fold and develop correctly. And interestingly, in these cases, we were able to also follow the embryos up through the larval stage, and they were able to hatch out correctly. So these tissues were all actually formed viable organisms afterwards. These embryos were formed viable. So what we basically are seeing, if we go back and look at the betweenness centrality in these networks compared at this time when tissue folding begins, that the perturbations diminish redundancy in the system, but ultimately some minimal path still exists across the tissue. So the remaining nodes are more important in connecting connecting cells together. So with damage, what we basically see is that the network becomes more and more like Boston, which is somewhat of a poorly engineered system, but still functions to some extent. So to conclude here, we see, interestingly, that the wild type embryo actually creates many more linkages than is minimally needed to fold the tissue. And so while that might be surprising because you would imagine it costs energy to form all of these network connections, it actually allows a tissue to be highly robust because different types of perturbations, which result in localized damage in the system or localized loss of connections. So for example, the genetic degradation or the laser hole ablations are unable, don't sort of remove all connectivity across your system, and some minimal connecting paths are still present across your tissue. However, if you actually do vertical ablation cuts, so you sever all spanning paths across the tissue, then the embryo actually doesn't fold and development can't continue correctly. So what we're seeing here is that redundancy in and of itself doesn't trigger tissue folding. So we wanted to understand, well, what does actually trigger the folding of the tissue? And so we hypothesized that maybe it's stress that needs to be built up in the network prior to tissue folding, where if the edges that are connecting cells together become sufficiently taut, the cells that are connected between them might start moving in a coordinated fashion, and this might lead to the global directed flow of cells that is seen during tissue folding, when they all start to sort of move together towards the cell midline, the tissue midline, and move down in Z.
Can you use a chemical to break up the myosin network?
What do you mean by that? Like,
I think you use another kind of molecule if you inhibit something. I mean, just more than genetic and mechanical. I mean, that looks like it.
Yeah. What was the reason why we did this particular perturbation is that you could get a range of phenotypes pretty well that had different levels of the network. So you got sort of more severe and less severe degradation of myosin, and so you had a large phenotype of actual networks that could form. The problem is if you do some things like if you inject drugs that's going to limit, for example, myosin's ability to accumulate, oftentimes like you disrupt all of the myosin and then you have no network at all and then the tissue doesn't fold at all. So here we use that because we could get pretty well sort of the interesting sweet spot between full disruption of myosin and kind of just apocalyptic death and the full myosin network. So I'm saying that maybe it's something about sort of stress or stiffness building up in these edges that are connecting cells that might trigger this coordinated movement. So what I mean by that is, like, you can imagine if two objects are connected with a floppy string, they will have largely uncorrelated movements. But if that string is pulled taut, they're now linked by something that's more like a rigid rod that's going to allow these two objects to move together. So we therefore quantified the straightness of our edges to see if they were getting potentially increasingly strained or straightened over time. And so to do that, we use this measure of tortuosity, which is the length along your edge divided by the distance r between the two endpoints. And what we can see here, looking at the average tortuosity of all edges in your network and looking at how it evolves over time, we saw that indeed these network edges became increasingly straightened leading up to tissue and folding initiation at t = 0. And so again, we asked, well, is there something important about this degree of straightness in initiating tissue folding? And so looking at how the straightness of network edges evolves over time in the mutants where myosin was genetically degraded to varying amounts, we saw that in these tissues as well, folding began when network edges also straightened to the same amount. We asked if there was a directionality in the straightening of these edges, because it's known that tissue, the direction of highest tissue tension is along the anterior or posterior axis, so parallel to this fold. By separating out edges that were AP oriented along the AP direction and separating out with those along the dorsal ventral direction. And indeed, the average tortuosity, or the straightening of AP fibers, was more significant than in the perpendicular direction. So to better understand what mechanical function this straightening or potentially stiffening of the fiber network could have on the folding of the embryo, we collaborated with Pearson Miller and Jorn Dunkel in the MIT Math department to make a mechanical model of the folding embryo. So the embryo is modeled as an elastic shell where myosin contractility is modeled by altering the preferred curvature of the shell in a rectangular patch on the shell surface, as you can kind of see here in red. And that causes it to bend, the shell to bend inward in this region. But then in addition, we imposed stiff elements on the surface of the embryo, which were modeled off of our myosin network data, where the stiff edges were adding an additional energy cost for the tissue or the shell to bend along the edge's length. So what happens? So first see what happens without this myosin network. Let me show you what happens just with the myosin contractility on the shell. So as I mentioned before, if we tune up the amount of myosin contractility, we go from the shell dimpling to forming this furrow shape, which is similar to what we see in experiments. But then if we add this stiff network in addition on top of our shell, what we see is that this furrowing shape, so here this green region, occurs at a lower level of myosin contractility than on a shell that does not have this stiff network. So basically these stiff oriented fibers are playing a structural role in robustly guiding tissue folding in despite varying degrees of myosin contractility or sort of motor levels. If we look at how important the orientation of these edges are in this effect, we can look at how the furrowing happens on the shell with shells that have no network, one that has a dorsally ventrally biased network, and one that has this anterior posterior biased network. And not just the presence of the stiff network, but actually its orientation along the AP direction is most favorable for a furrow to form fastest and deepest. So there'll be this yellow line here. And interestingly, if we go back and look at our experimental data when myosin is degraded, so when we actually have lower amounts of motor than normally in the wild type, we actually see an increase in these anterior posterior AP fibers in the remaining network present in the tissue. And that's interesting because that's basically the most, if you have a low amount of myosin, that's the best sort of use for your myosin is to arrange it in these AP fibers to get the most robust tissue fold. So basically to conclude, what we're seeing is two parallel strategies that encode intrinsic robustness via cell interactions. First, redundancies in the myosin network ensure that loss of multiple linkages is inconsequential for folding, and also stiff oriented fibers that play sort of a structural role in robustly guiding folding. And so this really highlights the idea that understanding the patterns of cell interactions in a tissue is critical to understanding tissue level behavior. And more broadly, well, I showed you the sort of supracellular network in one tissue context. They've actually been observed in many different developing tissues, and they've been really poorly understood. So this is just one example here, as I showed you at the early part of the talk, this neural tube formation in the mouse, so it's going to go on to form the central nervous system, also has these supracellular myosin linkages across many cells in the tissue. So moving forward, it's interesting to think about how these structures form and how they coordinate many different types of shape change events in 3D structures. I think just sort of more generally, what's also sort of an interesting question is to think about what actually determines network architecture and why some architectures are formed versus others. You can imagine, well, I said that this network is very redundant and therefore highly robust. Ultimately, the embryo forms a network with very few connections with its neighbors, around 2 or 3. But if you want to organize nodes into the best configuration for a robust system, you really want to form something that looks more like a scale-free network where you have some hubs that are very highly connected. So is there, in these sort of biological systems, is there a trade-off between robustness versus the cost to build this network that's at play here? And I think it's interesting when thinking more broadly about engineering different types of tissue shape and tissue shape change in a robust manner. So just to conclude, taking this tissue scale approach to decipher morphogenesis really has the potential for this new description of how tissues change shape that can be used when thinking about both reprogramming, shape of developmentally defective tissues, but also when thinking of to engineer new self-folding tissues and soft materials. And so as you're thinking about, can we now form materials that, during development, form these complex 3D structures, but maybe also have some of these interesting properties of living tissues where they can reconfigure, change shape, and evolve over time as well. So with that, I'd like to just thank Adam Martin and Pascal Sibelzon, whose work I did in their labs. Pearson Miller, I'm here, and thank you so much for your attention.
Great, thank you so much. Really interesting work. I think, and so then I think we're going to open this up for questions now, and Booble, I think, has the first question.
Okay.
I have a
Hi, so I am quite familiar with this active tension network model from Boris Schreiman and its predictions about the folding. How does that fit into, or is there any way you have tested that using your experiments or in the folding of the fruit fly?
Well, we haven't, so because in that situation, you have tension along the edge, right? So the nodes are being pulled together along connections between them, right? In this model, an active tension model that you're describing.
The active tension model is just a force balance model.
Yeah, so it's sort of like springs between nodes, right? And so that was actually the first direction we went with in this project, was trying to find a parallel between this network and something like the active tension model. But interestingly, what we were seeing is that sort of this more of a passive role of the supracellular network unfolding. So when we started modeling how more of an active network on a surface of an embryo might change the shape, I mean, you would imagine that the nodes are getting pulled together, and that would cause surface area reduction along the AP axis. But actually what we were seeing is that these edges weren't really pulling in that direction, but instead cell constriction occurs in the perpendicular direction. So that doesn't really fit with that description. So does that make sense? Basically, we see increased alignment of these fibers along the long axis of the embryo. the AP axis, but we're seeing constriction happening, cell area constriction happening in the perpendicular.
I don't think that's in any contradiction with the active tension network model at all. There is nothing saying it extends along that direction. Those are the soft modes. Your soft modes could be very different, but the whole active tension network model is just saying, I have force balance at every node, which you certainly do.
But the way that the nodes evolve over time, we didn't. I'd be happy to talk more about it. I guess it's a longer description. Yeah, but we started to model that it wasn't fitting with our experimental data. But yeah, maybe we were doing something wrong. So I'd love to talk more about it.
Thank you.
Yeah, but that was something we had sort of thought about, and yeah. And then we ended up seeing that ultimately it acts more like these rigid rods on the surface that aren't really pulling along the length of the rod. They just sort of act as this passive scaffold to restrict bending in one direction and promote bending instead along the perpendicular direction. But yeah, maybe we can talk more.
Okay, we can talk more. Yeah.
Yeah.
We have another question, this time from Yana, who stole my question, I guess. How is the folding encoded genetically? Do you think genes specify material parameters or something about the spatial distribution of protein components? In general, how do we bring the question of control of folding back to genes?
Yeah, well, there's definitely, genes that are encoding. So it's like you have twist and snail, which basically are patterned in somewhat of a gradient across this region, this ventral region, and that patterns downstream the appearance of myosin in this ventral region, so in this patch of cells. And so that defines things like, for example, the geometry of how many cells are going to express myosin apically and then start to connect together. It's also can impact the material properties it can impact, cells that don't have that are going to that don't have myosin are going to stretch more. So neighboring cells stretch more. The ones that have myosin become increasingly stiff. So yeah, a fair amount of work has been done on more of the gene aspects of it. And that's sort of why I went in the different direction, because there was still sort of a gap in thinking about how do those patterns, and then particularly the myosin, which is being sort of the control, what's really driving this fold, how is that sort of at the tissue scale impacting these movements? Yeah, but you could bring it back to, you could for sure bring it back to the gene level if you want to.
Yeah, I think there's a lot of interesting questions there, right?
Yeah.
As this thing remodels itself, then these patterns of genes that you showed in the first slide are remodeled.
Yeah. This is, for example, when I expanded the shape of the ventral region. So you can do that by inhibiting like ventral cell, something that basically tells which cells to become ventral and which cells not to be ventral. So you can play around with the gene patterns and then basically change, as we saw, the overall properties of the myosin network, and then the way the tissue folds.
We've got a question from Umida.
Yeah.
Hi, so I had a question. I had a question about the relationship between redundancy and robustness. Have you tried to make that a little bit more quantitative? And is it a monotonic relationship, or presumably there is an optimum range of redundancy, which will help with robustness, but too much redundancy not necessarily is going to help you?
Yeah, that I was in this system. You're pretty limited where you can't really get more redundant than the wild type. At least I hadn't been able to do that experimentally, so I was only at a place where I could degrade things. But I think that's definitely an interesting question. It was what I was trying to touch on at the end, where basically your cells are only connecting with about 2 to 3 neighbors, whereas they have, you know, a packing of around 6. And so it's an interesting question of like what would happen if they were connected to all of them, and would that change the properties of the tissue, or maybe that's too much redundancy. It is something in further directions you can start exploring in different tissues. I've kind of touched on the fact that different tissues have these types of networks. And in some contexts, you have networks, it seems like, with more connections with their neighbors. And so maybe it would be interesting to see if that defines different types of shapes, or maybe a more or less robust morphogenetic event. Yeah, but that's definitely interesting. I mean, there's a lot of different types of network properties you can imagine sort of calculating and seeing how they evolve.
Right, and I guess also depends probably on the kind of perturbations that you're going to, subject your network to, right? Like robustness with respect to what kind of properties and what kind of perturbations is probably also important.
Yeah, yeah. No, that's a good question.
Thank you, very good talk. Hey, Seth, I don't know, I see you raised your hand. Hello.
Fibers and by the track. And so the question is, 200 micron, the cells started to move more rapidly along the fiber. And you said that was due to this of these vortexes.
Mm-hmm.
Because the fibers are aligned in the direction of motion, and then because you have also the fibers would become aligned in these cells. So there's 2 things going on. They're moving faster down the fiber, and then they're being aligned. And for the small radius of curvature, the alignment of the fibers was perpendicular to the axis of the they were moving faster. So what correlation there between the motion and the orientation of the what lesson do we learn about mechanics of cell motion?
Yeah. So if I understand correctly, because you're cutting out a little bit, basically that at this very small radii, we were seeing the circumferential rings, but actually seeing movement in the perpendicular direction. And so you're asking how does that how do you explain that?
Yeah.
So that was surprising, actually, because we would have thought that it also, there's a result that I didn't show there, but these rings get, there's a increased adhesion of the rings with the substrate. So they actually have more cell adhesions with the substrate than cells that were on a flat surface. I don't know particularly why, I would say. It's not completely shocking. I mean, in like a migrating lamellipodia, you have arcs of actin that are perpendicular to the direction of motion. So here they're curved around the surface of the wire, but it's not, yeah.
I'm just used to the treadmilling in the direction of the protrusion,
Yeah.
but
yeah, I mean, you still saw protrusions that looked relatively normal at the leading edge of these collective groups of cells. So in that sense, they weren't probably didn't have the same architecture as we were seeing in the bulk with these rings. but I didn't
But-
look with high enough resolution to see if they were having any different types of organization.
And then it wasn't clear to me what the shape of the cell is when it wraps around the fiber. Is it elongated? In which direction is the cell elongated when it wraps?
Well, so what was interesting is that I have like kind of one reservoir where there's a lot of cells, and they start migrating out on these curved surfaces. And so what you have is you have a gradient in density across your wire. And what I saw is that independent of density and density usually impacts cell shape, is why I say this.
Okay.
So when it's less dense, cells are kind of wider and more spread. You always saw these rings. These cells normally remain relatively hexagonal, so they never adopted a very elongated shape in any case. What was interesting though is on very, very tiny wires, so I went down to like submicron size, you actually got very long protrusions that would form off the end of them. And cells, there would only be enough room for one cell to be on the wire, and they would actually wrap around the whole wire. So it'd be like a donut with a wire in the center, and then the cells would self-fuse on themselves. So you would really have like a donut-shaped cell with a wire in the middle. And then in that case too, you had rings. So it was all very consistent, but not very interesting in terms of what's actually happening with the migration, yeah.
Okay, interesting. And then in the furrows, what was the organization similar?
In the folding the embryo?
No, in the furrow, and you also did grooves, in the grooves.
Oh, in this work, I didn't do furrow. I didn't do negative curvature. I kept everything on the, oh, you mean the tracks, the 2D
Yeah,
tracks?
track. Yeah,
Oh,
track.
okay. So that was flat, and there, basically cells at the boundary of the track would align with the boundary of the track. But then the cells that were not next to the boundary would not have any preferential alignment. So basically, as the track got wider and wider, you kind of lost this boundary effect when you averaged across the entire tissue. And you saw, yeah.
All right, thank you.
No problem.
If there's no one else that has a question, let's thank Hannah one more time, and we'll see you next week on Thursday.
Thank you.
Thank you, Hannah.
Bye.
Thanks.